présenter.
Okay.
Bonjour. Donc je fais la présentation de Giovanni Fiorito parce que je le connais assez bien. Et Giovanni est un biostatisticien, bioinformaticien qui travaille à Turin à l'Institut Italian Institute for Genomic Medicine qui s'appelait avant Human Genetics Foundation où je travaillais avant de venir ici. Et donc Giovanni a été recruté quand j'étais là comme directeur de la fondation.
I don't understand French.
Ah oui, je regarde Giovanni, mais il m'a dit je ne comprends pas. Donc, je peux faire l'introduction en anglais si tu préfères. If you want, I can do it in
If you want. Just don't look at me if you
So I was saying that you were recruited, in fact, when I was the director of the Italian Institute of Oncology, of Genomic Medicine, that it was called before Human Genetics Foundation in Turin. And he's actually a mathematician by training, and then actually worked for a bank as a statistician. That's right.
Right. Yes.
And at some point he got bored working for a bank. Very soon, yes, actually. And it was we were lucky of that. In fact, that he decided, in fact, to start a career, an academic career in biomedicine as a biostatistician. So Giovanni has worked in the institution in Torino with Paolo Vines currently, and he has conducted a lot of work in the field of epigenetics, particularly on cancer, but not just on cancer, as he will show you today. I think that's it, and you can start to actually add that at some point more recently Giovanni started to work with Alexis and Fanny on studies on Parkinson's disease, and this is why also Alexis invited Giovanni this week to work with him. Please.
Okay, so thank you for the presentation, and thank you, Alexei, for inviting me again. So I will talk to you about some results of a project, a European project called Lifepath, that is focused on social inequalities in health, as I will show you in a moment. Okay, so this is the group Gianluca was talking about at the Italian Institute for Genomic Medicine. Our research combined epidemiology, so let me say classical epidemiology, in which one can study the effect of a risk factor on a certain outcome of interest, with molecular biology. That is the use of the so-called omic biomarkers. Omic biomarkers means all the large-scale biomolecular data that can be produced and analyzed thanks to the recent technologies. So omic refers to large-scale genomic, transcriptomic, epigenomic, that is what I'm going to show you, proteomic and so on. So we try to identify with this simplified model here some intermediate biomarkers between risk factors and a certain outcome. Okay, so I told you that Lifepath is focused on social inequalities in health. Alexei told me that most of you in this room are epidemiologists, so I'm sure I don't need to convince you about the first sentence here, so that low socioeconomic status is associated with lower longevity, high risk of chronic diseases, age-related diseases, but in general with a lower quality of life at older ages. So among the aims of Lifepath, we have these two key questions. The sociologists in our group like to use this expression here. So how low socioeconomic status goes under the skin. I mean, which are the biological mechanisms behind this association? And another important point is to try to understand when to intervene. That is, for example, for a policy intervention that want to reduce the social inequalities, in which period of life an intervention could be more effective in terms of reducing inequalities in health and improving the quality of life of individuals in the lower social class. Okay, so to study the healthy aging, we need first of all to define it, and we use this definition here. Healthy aging is the maximum state of functional, physical and mental well-being that can be reached in any particular stage of life. And we use this model of physical functioning as a function of age. So as we can see here, we have in the early stage of our life, we have an increase in the physical and mental functioning, and then after a certain age, the decline phase starts, and we have a gradual losing of physical functioning. And this model here indicates different trajectories of this physical functioning during life, and show that health inequalities begins very early in the first years of life, maybe even before. I mean, because all of us have inherited from our parents some genetics, epigenetics and behavioral factors that can affect our health during all the life course. And another assumption that we have in the Lifepath project is that is based on the observation that social inequalities in health constantly increases during aging. So we thought that the early stage of life is very important also for the decline phase. So our idea in Lifepath is, wow. So, okay, someone can help me. Just touch maybe.
No, maybe.
No? I touch. Ok, it was me.
Annuler, appuyer sur une autre touche.
Non, pas de signal.
Ça a coïncidé avec un geste de la main.
Putain, mais c'est bizarre, hein. Peut-être que j'en mette comme putain. Mais on n'a rien fait.
Essaie de l'étendre vers elle.
Okay.
Okay.
Thank you. I will not touch the. Okay, so I was telling you that okay, if we want to study healthy aging and the differences in older ages, we need to study all the life course. And in Lifepath, we want to. So we think that all the biological changes that are related to some exposure, social, environmental exposure, can be traced using these omic technologies, so the biomolecular measurements. Okay, this is a representation of the cohorts in Lifepath. We use both Europe-wide or national survey data, but mostly prospective cohort studies. We have different collaboration in Europe, in Italy, in France, in Portugal, mostly Great Britain. Also one cohort in the US and one in Australia. Overall, we have around 250,000 individuals with a biological sample already archived, and among those 50,000 individuals have already a biomolecular measurement, one of the omic technologies that can be analyzed. This is another representation of the cohorts in Lifepath. This is just to show you that for some cohorts, for example, young things, we have data from birth to adulthood. From others, we can cover, for example, tilde. We can cover all the decline phase. Well, this picture actually is to show that we can study the effect of a risk factor of low socioeconomic status, in our case, across all the life course of an individual, simply putting together all the results like pieces of a puzzle. Okay, this is another representation of the cohorts in Lifepath, the last one. This is from the website of Lifepath. If you go on this link here, this page is actually an interactive page. So if you want to know so in which cohort you can study a certain outcome, you can just click there. Let me try. Okay. If you click here on cancer, you can see in which cohort we have this kind of data. And the same if you click on a cohort, E3N, or E3N in French. Okay. If you click here, you can see which outcome you can study here and with which biomarkers, and the same for the biomarkers. So if you click on a biomarker, you can see in which cohort we have this data. And this tool, I think, is very useful for us because when we think on a new project, we can immediately know which kind of the quantity of data we can use. But I hope that could be useful for you also, if you are interested in some collaboration on certain outcome. You can take a look at here, and if you are interested, please let me know or write to Professor Paolo Vinis, that is the coordinator of the project. Okay, so these are two popular results in Lifepath based on epidemiological studies without biomarkers. The one on your left, yes, left, is about the association of low socioeconomic status with mortality. And what is interesting here is that the effect of low socioeconomic status is actually comparable to those of other risk factors like physical activity, alcohol intake, hypertension. Only smoking and diabetes have a stronger effect on mortality compared to low socioeconomic status. And The second study here is about physical functioning that is measured with walking speed, as you know. Again, the effect of low socioeconomic status is comparable, both in men and women, is comparable to those of other risk factors, obesity, diabetes, and so on. In fact, in this paper, our colleague Silvia Stringini suggests that low socioeconomic status should be listed in, should be put in the list of the 25 per 25 risk factors. Okay, so I will talk you about DNA methylation biomarkers. So just a brief introduction on epigenetics. So with epigenetics, we refer to all the molecular mechanisms that regulate gene activities, so determine which gene should be active and which other should be silenced, without any change in the DNA sequence. So there are three main epigenetic mechanisms. So histone modification, DNA methylation, and non-coding RNA regulation. And in this representation here, there is just an example on how epigenetics could regulate gene activity. So, for example, here you can see that the DNA is closed around the histones. So in this case, the transcription factor cannot bind the DNA. So the transcription of the gene that is around here cannot start. Instead, when the DNA is open, like here, the transcription factor can bind the DNA, so the transcription of the gene can start. And these mechanisms are regulated by one of these three processes mainly. An important thing to know about epigenetics is that it is cell-specific. That is that each cell of our body has its specific epigenetic profile. And this is the reason why different cells in our body have different function. So for example, if I take a cell of my skin and a cell of my hair, they have exactly the same DNA sequence. But the genes that are expressed in my skin and the genes that are expressed in my hair are not the same. So this differentiation is related with epigenetics. Okay, so I told you that DNA methylation is one of the epigenetic mechanisms. So it can appear in each place in our genome in which there is a cytosine followed by a guanine. So these places are called CPG sites. And in CPG sites we can have, in fact, non-methylated cytosine or methylated cytosine. So the mechanism is just that a methyl group bind the DNA molecule. And this regulates the transcription. So usually when the DNA is not methylated, the transcription factor can bind the DNA and the transcription can start. That does not mean that will start, because could be that there are other mechanisms later on, but at least the transcription can start. Instead, in the second case, here when the DNA is methylated, the transcription factor cannot bind the DNA because of biochemical reactions, and the transcription cannot start. Okay. So why it is interesting to study DNA methylation? Well, because it changes whatever we do, actually. It's not fixed like the DNA sequence, but react as a function of external and internal exposures. So it changes with diet, with physical activity, so whatever. Okay, this slide is about how to measure DNA methylation, how to measure full genome DNA methylation. So a lot of a lot of CpG sites in our genome. And the two main methodologies are microarray. So the most famous is the one we use, the Illumina bit chip microarray. And the second methodology is next generation sequencing. There are pros and cons. So, for example, according to my colleagues, the lab technicians, the microarray is quick and easy to do, to perform the experiment. From a statistical point of view, from a bioinformatical point of view, the pipeline for pre-processing and data analysis are already established. So, because it is a technology that has actually 5 to 10 years. It is cheaper, so microarray is cheaper than next generation sequencing, and requires less quantity of DNA. Sometimes it's not easy to store a higher quantity of DNA. So these are the advantages. On the contrary, if we use microarrays, we have information on just a subset of CpG sites in our genome. With the Illumina microarray, they are about 850,000. Instead, with next generation sequencing, we have all the genome, all the CpG sites in our genome. And an important point is that with next generation sequencing, you can control for the effect of genetic variants that influence DNA methylation, because when you do NGS, you also have the genetic information. Instead, with microarray, you cannot have this control. Okay, so whatever you use, in each case, the number, the numerical value that you will analyze is the so-called beta value, that is a proportion between 0 and 1. So I told you before that methylation is a mechanism that is 0-1. So a cytosine could be non-methylated or methylated. So one should expect this kind of data, 0-1. But what we measure is, I'm touching again something, what we measure is a mix of cells. So the beta value represents the proportion of methylated cells from that specific point in the genome. Okay. Okay. This slide is about the relationship between DNA methylation and aging. That is very interesting. DNA methylation is constantly changed during aging, as I told you before, as a consequence of exposures, but also naturally, let's say. There are two main mechanisms of changes in DNA methylation during aging. From one side we have the epigenetic clock, the so-called epigenetic clock, and from the other side we have the epigenetic drift. So the epigenetic clock refers to specific point in the genome, fixed, in which DNA methylation constantly increase during aging or constantly decrease. And this point in the genome can be used to predict with a very high accuracy the chronological age, as I will show later. Instead, the epigenetic drift is the tendency of increasing variability, increasing discordance between epigenomes. This example, I think, is very clear. These are two pictures of chromosomes of two identical twins, monozygotic twins, at 3 years of age and at 50 years of age. And the yellow part represents the point in the genome where the two identical twins have the same DNA methylation tag. Instead, red and green represents the point in the genome where they differ. So this means that at the beginning of our life, they were almost identical, epigenetically identical, and then there was a divergence in the epigenomes, the so-called drift, epigenetic drift. Okay, so coming back to the epigenetic clock, in 2013 came out this very popular paper by Steve Horvath that showed that you can actually use about 350 CPG sites to predict with a very, very high accuracy the chronological age. Here on the x we have the chronological age, and on the y-axis the predicted age using these 350 CPG sites. Also, he introduced, Steve Horvath introduced this concept of epigenetic age acceleration. That is simply the differences between your epigenetic age, that he called also biological age, the differences between epigenetic and chronological age. So, for example, here an individual here have a chronological age of around 60 and an epigenetic age of around 70. So this means that this guy here is aging faster, and the same on the contrary, if an individual have a chronological age that is lower than, no, a biological age that is lower than his chronological, means that he's healthy, is aging in a good health. And after this paper, a lot of papers came out that shows that epigenetic age, or the age acceleration measure, is a very strong predictor, first of all, of mortality. So individuals that have higher, that actually experience epigenetic age acceleration have a higher risk, have a lower longevity. And this picture is from a very nice review about the field that actually showed that also age acceleration is associated with cancer, with neurodegenerative diseases, is associated also with some risk factors like obesity, infections, and the good news is that you can slow down your clock by an healthy diet and physical activity. The second plot here is another review. This is from Steve Horvath himself, that actually compare different epigenetic clocks, because there are other measures of epigenetic age acceleration. One is from Annum, that is specific for blood and actually works even better in predict mortality, and another is more recent from Levine. This review actually show how to interpret different clocks. The natural question now for Lifepath is what about socioeconomic status? It's associated with age acceleration, and this is exactly what we have done. So we try to investigate this association, and we also try to answer this questionnaire, so whether early life socioeconomic status has a stronger effect compared with adulthood socioeconomic status. For this study we collected DNA methylation data from blood samples from 3 cohorts with an age range from 35 to 80, so the decline phase, and we ran cross-sectional investigation on the association of SES with age acceleration. So these are the outcomes that we used, both the Horvath and Annum age acceleration. The predictors were proxy for socioeconomic status. We use education, categorized in 3 categories, and then we use another proxy that is based on occupational position that try to simulate life course trajectory. So we use father occupational position as the proxy of childhood socioeconomic status, and the household occupational position, highest occupational position, as the proxy for adulthood. So we have 4 categories. So II, that is the reference, the 2 intermediate categories, and the worst situation. And we use linear regression models to investigate this association. Okay. These are the results. So these are the estimate adjusted for possible confounders and mediators. And as we can see here, there is gradual increase of the age acceleration from high to low socioeconomic status. Also here we can see that the effect of childhood socioeconomic status is greater than the effect of adulthood. And since these 2 intermediate categories have intermediate outcomes, this suggests that the effect could be at least partially reversible. Okay, this was the first results. Then I moved back again to the epigenetic drift. So epigenetic drift, the mechanism I described before, leads to the accumulation of epimutations. An epimutation is an abnormal repression of a gene that instead should be active, or vice versa, an activation of a gene that should be silenced. And caused by the epigenetic drift, these epimutations appear randomly in the genome and during life. And that's why they are called stochastic epigenetic mutations. A first result is that there is a strong association of the total number of stochastic epigenetic mutations with age. There is actually exponential increase during aging of these epimutations. And this is a process that occurs naturally. But also, well, first, this plot here is just to show that in 3 different cohorts in Lifepath we have replicated the exponential association. Here the trend looks linear, but the y-axis is on logarithmic scale. We also note that there is a lot of variability between age groups. And also recent literature showed that total number of epimutations is associated with cancer risk factor like alcohol intake and smoking. It's also associated with X chromosome inactivation skewing in women. That is a pathological condition that in turn is again associated with cancer and is frequent in older women. And also in cancer tissue, another study showed that cancer tissue compared to adjacent non-healthy tissue has a higher number of epimutations, and in hepatocellular carcinoma, the number of epimutations correlates with tumor staging. So, seems a process that increase with cancerogenesis. So again, we ask, what about the association with socioeconomic status? So we have investigated this association, and also we have tried to understand which are the other determinants of the variability in the number of epimutation across individuals. Then we have investigated the association with cancer, and finally we try to understand whether these epimutations are really randomly distributed in the genome or instead are enriched in functional genomic regions. Okay, so for the first part of the study we collected around 15,000 DNA methylation data from blood, and we have investigated the first part, that is the association of socioeconomic status with epimutations with a cross-sectional study design. Instead, for the second part about cancer, we have data from 3 prospective cohorts in Italy, Australia and Norway, with a matched case control study design nested in the cohort. So the blood sample was collected when they were at recruitment, when they were healthy, and then we designed this case control study respectively. And we studied the association of epimutation with 8 different types of cancer. Okay, so first of all, well, this is just to show you that we have harmonized all the covariates because of different sources of information. For example, alcohol intake and physical activity were collected in different ways in the different cohorts. So we decided to treat all the variables as categorical with 3 categories. And in this way we can also compare the effect of each factor with those of socioeconomic status. Okay, this picture is about the statistical method to compute this total number of epimutations. So basically for each probe we look at the distribution across all the individuals and we identify the extreme outliers of the distribution. So here is just an example on 20 CPG sites, but we do it genome-wide. For example, sample number 1 has an extreme outlier in probe 2, another here, another here. So he has a total of 3 epimutations and so on. So our outcome of interest is the total number of epimutations that are defined in this way. Extreme outlier means the DNA methylation is lower than the first quartile minus 3 times the interquartile range, or higher than the third quartile plus 3 times the interquartile range. Okay, so our outcome is the total number of epimutations. We use the logarithm transformation because of the exponential relationship with age, and we run different linear regression model, one with all the adjustment for lifestyle variables and some technical and cohort specific variables, and the second model includes for each risk factor, for each lifestyle variable, an interaction term with age to see whether the effect is age dependent. And finally we run a random effect meta-analysis, and these are the results about the comparison of the two extreme classes. So high CES was the reference and low CES has a higher number of, so individuals in lower classes have a higher number of epimutations, around 6% more. Okay, this is the comparison of the effect of low socioeconomic status with those of other risk factors. Again, we can see that the effect is comparable to that of other risk factors like alcohol and physical activity, so according with the results on mortality, and smoking has a higher effect. And this part is about the models with interaction with age. As you can see, for each of the risk factors there is a significant interaction. That means that the effect of a risk factor is higher in older individuals. So putting all this information together, we can say that the total number of epimutations could be considered as a good biomarker of exposure-related damage during aging. This is about the second part of the study, about the association with cancer, just a description of the study sample. They are about 5,000 case control pairs with 8 different types of cancer. And we use this time logistic regression model because our outcome is case control status. We run a minimally adjusted model and a complete adjusted model to double check whether the effect of epimutations depends on other risk factors. And finally, for the studies that we have in more than one cohort, we run again meta-analysis. And these are the results. So we found a significant association for breast cancer and for lung cancer, and in MCCS also with mature B-cell neoplasms. This association is very strong, and I think this result is not surprising because we are looking at epimutations in the blood. This is a cancer of the blood. Okay, so the last part of the study is about the analysis of the distribution across the genome of these epimutations. So without going into the details about the bioinformatic tools I used, I compared the distribution of epimutations with a list of functional regions defined by the ENCODE project. And what I found is that actually these epimutations are not randomly distributed, but are enriched mostly in transcription factor binding sites of two famous proteins, EZH2 and SUZ12, that are two members of the polycomb repressive complex. This is a group of proteins that have a very important role in cancer because they are responsible of tumor suppressor regulation. A very interesting result is that the same enrichment was found comparing cancer tissue with healthy tissue, the study I told you about before on breast cancer. Okay, so basically what we found is that there is a mechanism that is deregulated in cancer, and we were able to see the same enrichment in the blood of healthy individuals years before the cancer appearance. So this suggests us that this could be a mechanism of early stage carcinogenesis. The mechanism is this one that was described in cancer. So this is a tumor suppressor gene that should, if the cancer is not present, should be suppressed. And in normal condition, the repression of this gene is done by polycomb. So when an epimutation appears, so these points here from non-methylated become methylated, the regulation by polycomb go away, and the suppression, the regulation of the gene is done by DNA methylation. The point here is that in normal, in healthy people, the gene, the tumor repressor gene should be suppressed. But in case of cancer appearance, this mechanism of regulation is more plastic. So if the repression is done by polycomb, in case of appearance of cancer, it can react. So polycomb can go away and the tumor suppression can be activated. In the second case here, if the suppression is done by DNA methylation, the process is actually more fixed, it's not reversible. So in case of cancer appearance, the tumor suppression could not be activated. So the tumor progression can start. I don't know if I was clear. It's quite difficult to explain even in Italian, so ask me if I am not clear. By the way, this is another advantage of using biomarkers in epidemiology, because in this way we can hypothesize mechanisms of carcinogenesis. And we are thinking about some kind of in vitro experiment to confirm this hypothesis. Just a note, these two proteins, the targets of these two proteins are enriched of tumor suppressant genes, but also for genes related with neurodegeneration. So that's why I ask you and Fanny to try to see whether there was the association with Parkinson. Okay, these are very preliminary results from a study of new birth. Generation 21 is a cohort of newborns. This is the association with socioeconomic status of the mother with epimutation of the child, and very preliminary, but it seems that there is an association with a higher number of epimutations at birth associated with lower education and lower income. So take it with care because very preliminary again. Okay, that's it. This scheme is just to summarize what I told you. So I show you that low socioeconomic status is associated with age-related DNA methylation dysregulation, that in turn are associated with lower longevity and higher risk of cancer. And this association is true basically for any stage of life, from birth to adulthood. The effect seems to be stronger in the early phase of life compared to the decline phase. And okay, so this was our first time we have described a biological mechanism to explain how low socioeconomic status goes under the scheme. These are some projects ongoing. We are investigating, as I told you, the effect on children cohorts, and also we can try to see whether there is associations with other outcomes, cardiovascular disease and so on. And finally, from the bioinformatic part of the project, we can try to see whether we can identify some specific signature for each type of cancer, and maybe also for our project, Alexi. Okay. Okay, that's it. So just to thank you all the participants of Lifepath. They are too much to list out the names. Again, thanks to my group, and thank you all for your attention.